Hello friends, welcome back to my YouTube channel Nesting Padasala. This is a part 3 video. Currently we are revising for the DBT BITP examination. So please watch the past 2 videos which I have published. So please read the disclaimer that I have given in the description below. So first question, which substitution matrices should you prefer to find the distantly related orthologs through blast search? Okay, so Blossom matrices, which means block substitution matrices, and PAM matrices, which means point accepted mutation. Okay, don't go in depth about studying these kind of matrices and all. So Blossom matrices with high number and PAM matrices with low number are designed for comparing closely related sequence. Like Blossom 90, that is Blossom matrix for high number, and PAM 1, which is PAM matrix with low number, are generally employed for more conserved and less divergent sequences. Like if you are going to compare the amino acid sequence of rat versus mouse, you can go for Blossom matrix with high number and PAM with low number. Okay, whereas Blossom matrices with low number and PAM matrices with high number are designed for comparing distantly related proteins. Say Blossom matrix of 45, which is low number, and PAM matrix of 250, which is high number, are generally used for comparing more diverse sequences or less conserved sequences. That is, if you are going to compare the amino acid sequence of rat versus bacteria, means you can go for Blossom matrices with low number and PAM with high number. So here they are asking about distance, then you should use Blossom matrices with low number and PAM with high number. So option A is correct. Next, vaccine currently used in India's pulse polio campaign. So this particular polio campaign was launched in the year 1995 in order to eradicate the polio from India, and they had used oral polio virus vaccine. So vaccine for polio virus are classified in two types. One is oral polio virus vaccine and inactivated polio vaccine. Okay, so oral polio vaccine, they contain attenuated or weakened version of the polio virus, and for this particular India's pulse program, they had used OPV, which is live and attenuated. So correct answer is option number C. Next is inactivated polio vaccine, which was developed in the year 1955 by John Salk. So it is also called a Salk vaccine. It consists of inactivated or killed virus. Okay. Next is Trypanosoma brucei complex, transmitted to man by bite of several species of tsetse fly. It can cause sleeping sickness. Okay, so here you can able to see Trypanosoma brucei is responsible for causing African sleeping signal, which is transmitted by the bite of tsetse fly, and Trypanosoma brucei is responsible for transmitting Chagas disease, which is transmitted by the fecus of kissing bug. Okay, next is inosine or inosine in the tRNA of anticodon will base pair with all except which of the following bases in the codon. So here if you check with the diagram, the green color is your mRNA, which will be carrying a codon, and this orange color is your tRNA, which will be carrying an anticodon. The third base in your mRNA and a first base in the anticodon of tRNA can form orbital base pairing. They follow a usual base pairing, at the same time they can go for base pairing with some other bases. Or else we can say the base that is present in the 3' end of their mRNA and the base that is present in the 5' end of the anticodon of the tRNA can form orbital base pairing. So here if you check inosine, which is present in the first position of anticodon, it can form base pair orbital base pairing with adenine. At the same time, it can form base pairing with uracil. At the same time, it can form base pair with your cytosine. It cannot able to form orbital base pair with guanine. The other example include guanine, which can base pair with uracil. At the same time, it can also base pair with cytosine. That's a usual Watson and Crick base pairing. And uracil, which can form base pair with adenine. At the same time, it can also form a orbital base pair with guanine. Next is DAPI, which is 4,6-diamidino-2-phenyl-indole. It's a fluorescent which is used for staining. This DAPI stain is used for both for your DNA as well as RNA, but it can be able to use for protein. This DAPI has a capacity to generally binds to AT-rich regions of your double helix DNA. At the same time, it can also bind with the RNA, but the fluorescent signal will not that much strong when you compare with DNA. Okay. Next is COAS, which is a bis-benzenamide dye. It has an intercalating tendency to bind with AT-rich regions of the DNA, just like a DAPI, and both the DAPI and this particular H strain are cell permeable, so they can be used for both for fixed cell and at the same time they can be used for living cell. Okay, but when you compare with DAPI, this particular bis-benzenamide, they have a low toxicity to DNA. Next is propyridinium iodide, which is membrane impermeable DNA stain. It is used to differentiate between living cell and dead cell in cell culture because this particular PI dye can't able to enter into the intact cell. Okay, and this propyridinium iodide, they are also intercalating agent, but they don't have any such kind of binding preferences. Like if you have DAPI, it specifically binds to AT-rich region, and this benzenamide, they also bind with AT-rich region. But when you take propyridinium iodide, they don't have any such binding preferences, and for protein staining, they will be using Kumasi brilliant blue. Next is measles, mumps, rubella is a MMR. Combined vaccine represents which one of the following vaccine category? So here I had this a Nature article. Here you can able to see measles, mumps, rubella, yellow fever, influenza, oral polio, typhoid, Japanese encephalitis, rotavirus, BCG, varicella zoster. Okay, all these diseases are protected by employing live attenuated vaccine. So the correct answer is option number B, live attenuated. Whereas if you take this hepatitis A vaccine or rabies vaccine, they are example of killed whole organism vaccine. And the example of toxoid vaccine include diphtheria and tetanus. So toxoid is a toxin protein which will be produced by the bacteria. So if you take diphtheria, which is caused by Corynebacterium diphtheria, which will be producing a toxin called diphtheria toxin, and tetanus will also produce a tetanus toxin. Okay. Next is subunit vaccine, which include either purified protein of bacteria or it include polysaccharide or peptide. The example include influenza, pertussis, hepatitis B virus vaccine, meningococcal vaccine, pneumococcal vaccine, typhoid vaccine, hepatitis B A virus. So these diseases are protected by employing the vaccine type called subunit vaccine. The example of virus-like particle vaccine include human papilloma virus, where they will be employing a human papilloma virus which don't have any capacity to cause disease. Okay, that's why it is called as virus-like particle. It only include a spike protein alone, and the example is human papilloma virus. And outer membrane vesicle vaccine, it is employed for group B meningococcal, which is a gram-negative bacteria, which will be employing this vaccine will be carrying a outer membrane along with a pathogenic agent alone. Next is protein polysaccharide conjugate. The example include Haemophilus influenza type B and typhoid, pneumococcal, meningococcal. Next is virus vector vaccine include Ebola, where they will be taking a viral vector. At the same time, they will be employing a viral vector gene and they will be incorporating a pathogen gene by Ebola from Ebola. Okay. Next is nucleic acid vaccine, which is example is a COVID vaccine, which is an mRNA vaccine. Okay, so here I had given first live attenuator was given for smallpox and killed whole organism was first introduced for typhoid, and if you take subunit, it was first given in the year 1917 for curing anthrax, and virus-like particle was first initially given for hepatitis B, and if you take viral vector, it was given in the year 2019 for curing or for preventing the outbreak of Ebola, and this nucleic acid vaccine, that is mRNA based vaccine, is given for SARS, and it was first introduced in the year 2020 for SARS-CoV-2 virus. Okay, next question is which one of the following modes of inheritance is seen in cystic fibrosis? Okay, so here I have given an inheritance pattern. So autosomal dominant, the disease that is following autosomal dominant pattern include Huntington disease, neurofibromatosis, onchorodoplalsia, familial hypercholesterolemia, Marfan syndrome. They will be following autosomal dominant pattern, and the disease that will be following autosomal recessive include Tay-Sachs disease, sickle cell anemia, cystic fibrosis, and phenylketonuria. Next is X-linked dominant, which include vitamin D resistant rickets, Rett syndrome, ornithine transcarbamylase deficiency, and the example of disease that is following X-linked recessive include hemophilia A, DMD, Fragile X syndrome. Next example or few traits that is following Y-linked inheritance include hypertrichosis in ears, webbed toes, porcupine man, and werewolf syndrome. Sometimes you will be noticing few man, they will be having a body hair in all regions. Okay, so these traits are following Y-linked inheritance. So the genes for all these traits will be located in the Y chromosome of male. Next, these diseases like Leber hereditary optic neuropathy will be following mitochondrial inheritance pattern. Okay, so here they are asking about cystic fibrosis. It is following a autosomal recessive pattern of inheritance. Okay, next is Huntington's disease is caused by. Okay, there is no such dinuclear repeat disease was not there. Huntington disease is a trinuclear repeat disorder. So the question is that whether it is caused by the repeat which is occurring in non-coding region or coding region. So here I have given you enough amount of explanation of the most common trinucleotide repeat disorder where there will be abnormal expression of your repeat sequence or microsatellite. First thing is your Huntington disease. The repeat sequence is CAG. For normal person, there will be repeats will be ranging from 10 to 30, but for the person with this particular Huntington disease, he will be having a repeat sequence that is repeated for 100 times. Therefore, it is creating a disease, and this particular repeat is present in the coding region, that is present in the HTT gene, that is coding region of HTT gene, which will be coding for this Huntington's protein. Okay, next is SBMA, that is spinobulbar muscular atrophy. The repeat region includes CAG. So for Huntington, spinobulbar muscular atrophy, spinocerebellar ataxia, the repeat sequence is same, that is CAG. Okay, so all the three diseases are classified type 1 repeat disorder because the CAG will be coding for glutamine. They are also called as polyglutamine disease. And for this particular spinobulbar muscular atrophy, the CAG repeat sequence will be present in the coding region, that which is present in the exon 1 of androgen receptor gene. Next is spinocerebellar ataxia. The repeat sequence is CAG. It is present in the coding region. As we take ferritic ataxia, the repeat sequence is GAA. Okay, so it is present in the non-coding region. That is, this particular repeat sequence will be present in the first intron of FRDA gene, which will be coding for a protein called ferrataxin. Okay, next is myotonic dystrophy type 1. The repeat sequence or the region that is repeated more time is CTG, which is this repeat sequence is present in the non-coding region of DMPK gene. Next is Fragile X syndrome. The repeat sequence is CGG, which is present in the non-coding region of FRN1G. Okay, here I have given you an inheritance pattern. Only the ferritic ataxia is following an autosomal recessive. Rest like Huntington disease, spinocerebellar ataxia, and myotonic dystrophy, they are following an autosomal dominant pattern. Whereas two diseases are following an X-linked pattern. Fragile X syndrome, which is following X-linked dominant pattern, and spinobulbar muscular atrophy is following an X-linked recessive pattern. Next thing, character regulated by Hollandrick gene. So Hollandrick genes are the gene that is present in Y chromosome. So generally males are having Y chromosome. So these genes are transmitted from the father to the son, and from the son it will be transmitted to his grandson. So first option is correct. Okay, it is passed from father to his son, then from his son, that is from his father, it will be transmitted to his grandson, because this particular gene generally affects only male, because this gene is located in the Y chromosome. Next is dental plaque is example of formation of, it is an example of formation of biofilm. Sometimes when you watch your teeth in front of your mirror, you're able to find a yellow color deposition in your teeth. Okay, it looks sticky. When you take them with your hand, it will be sticky. Okay, it will be just form a coat around your teeth. It is nothing but it is loaded with bacteria. So biofilms is nothing but they are the aggregates of microorganisms that will be embedded in a particular. It will be embedded either in a biotic surfaces like your teeth, or even in abiotic surfaces also they form an aggregation. So they generally get attached towards each other by producing. They will be producing a self-produced matrix of extracellular polymeric substances. Okay, Vibrio cholerae is generally employed as a model organism to study biofilm, and this Vibrio is responsible for causing the waterborne diarrheal diseases. So since this Vibrio cholerae has a responsibility to produce biofilm, it can protect themselves from the attack of environmental stress. Okay, so by forming a biofilm, it can form a protective covering, so it can be resistant by the attack by some other organism that can easily kill them. Okay, so we'll be continuing studying other diseases in the next part of the video, that is part 4. Thank you friends, thanks for watching this video.